Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing CSIR PYQs of the year 2013. I will be discussing the applied biology and methodology section. This is part 1 of the video. If you are new to my channel, please do like, share and subscribe. Then next one. Which of the following genes was engineered in the flavor saver transgenic tomato variety? So it is polygalacturonase gene that was used for flavor saver transgenic tomato development. Then for next one, for developing transgenic mice, embryonic stem cells are engineered to express the transgene. These cells are selected by neomycin. Neomycin is the selection agent. Then next one. In case of monoclonal antibody production by hybridoma technology, myeloma cells used lack the enzyme hypoxanthine guanine phosphoribosyl transferase or HGPRT, such that fused cells can only survive when selected on HAT medium, that is hypoxanthine aminopterin thymidine medium. What is the role of aminopterin in this medium? So aminopterin, it is used for blocking the nucleotide biosynthetic pathway. So the answer here is option 2, to block the pathway for nucleotide synthesis. These are all direct questions, that's why I am not going much into explanation. Then next one. In order to determine the primary structure of an octapeptide, amino acid composition was determined by acid hydrolysis, that is A. This one. This is amino acid by acid hydrolysis. The intact oligopeptide was treated with carboxypeptidase as in B, chymotrypsin as in C, trypsin as in D, and CNBR, that is cyanogen bromide, in E. The peptides were separated in each of the case and acid hydrolysis was carried out for B to E, resulting the following results were obtained. The brackets represent mixture of amino acid in each fragment. So first we will see the cyanogen bromide case. We will start from the end. So CNBR, it digests the carboxyl side of methionine residues. So if you see carboxyl side of methionine residue. So if you see, which one is that? E. Methionine is present in this half, whereas it is not present in alanine and serine. So methionine should be present at the end over here. Okay. So you will get the end of the sequence as methionine, alanine, serine, or serine, methionine, alanine, or alanine, serine, methionine. Sorry, it will not, methionine will not come in the middle. It will be methionine, alanine, serine, or methionine, serine, alanine. Either of these three combinations should have come. So if you see the options, you can see that methionine, serine, alanine is more common than methionine, alanine, serine. So the sequence at the end should be methionine, serine, alanine. That is at the C-terminal end. Then next we will see for the option D, that is trypsin digestion. Trypsin, it digests, as you know, at the carboxyl side of arginine and lysine. So here it will digest. This lysine would have been present over here. So it would have digested that, and then you get alanine, methionine, serine peptides. So trypsin digestion, before methionine it should be lysine over here. So like this, lysine, methionine, serine, alanine. So lysine, okay, it comes in two options, that is 3 and 4. So we will see for condition of 3 and 4. Then next one is in the case of option C, that is chymotrypsin. So chymotrypsin, it digests the carboxyl side of aromatic amino acids. that is WF Y. So phenylalanine is present over here. So it should have digested over here. So we got till lysine, methionine, serine, alanine. Now comes the turn of phenylalanine. Phenylalanine would be present before lysine. So phenylalanine, lysine, methionine, alanine, serine. So that comes in option B, and your answer is option 3. The next will be alanine, arginine, serine. Answer is option 3. Then next one. In order to study the role of telomeres in DNA replication, genetically engineered mice were prepared where the gene for telomerase RNA was knocked out. So telomerase, it is needed for during DNA during mitosis or meiosis, where the ends of the chromosome has a tendency to reduce in length with constant replication. So what the telomerase do is it act as a savior of the ends of the DNA fragment, and it will maintain the length of the chromosome during constant replication. So in order to study the role of telomeres in DNA replication, genetically engineered mice were prepared where the gene for telomerase RNA was knocked out. So what they have done here is to study the role of telomeres in mice, not in human beings. In mice, they knocked off the telomerase RNA. What happens, we will see. When cells from these knockout mice were taken and cultured in vitro, they proliferated even after 100 cell division, which is quite unlikely in the case of human cells. Human cells, I told you that already telomerase RNA is present, and the human cells have got a tendency to reduce the length of the DNA upon each cell division. So in order to prevent that, telomerase is active at the ends of the chromosome. So they prevent the chromosome from shortening in length after each cell division. So when the cells from these knockout mice were taken, but the knockout mice, they also expected that after 100 cell divisions in vitro, they will die off because the chromosome ends would be deteriorated, will be or shortened in length. So which of the following is the correct reason why this has taken place? Human and mice are fundamentally different with respect to their requirements for telomerase enzyme in the context of DNA replication. That is not correct. The telomerase enzyme, it is having the same function in both human and mice. Then in vitro, mice DNA becomes circular due to end-to-end chromosome fusion and does not require telomerase for DNA and replication. That is not so. They have not told anything regarding the chromosome ends becoming circular. Then mice have very long stretch of telomere DNA sequence compared to that of human. That may be one reason. That mice is having a more lengthier telomere DNA sequence than compared to the human. That may be one reason. In vitro, mice DNA replication does not require the removal of RNA primers. Okay, that is also not correct. So the answer is option 3. Next one. A non-enzymatic viral protein X was found to be inducing a cellular gene promoter activity. So the protein X, it was found to activate the gene promoter. Although no in vitro DNA binding activity could be identified with X protein. So they have conducted EMSA, that is electrophoretic mobility shift assay, for knowing the binding between the protein and the DNA, and they found out that the DNA binding activity could not be identified with X protein. So X is not a DNA binding protein as per the experiment. Then what they did next, it was found to be co-recruited on the cellular promoter along with a cellular transcription factor in vivo, but under in vivo conditions. That is when they conducted, I can say, a chromatin immunoprecipitation assay that is conducted for in vivo detection of protein and DNA binding or protein binding to DNA. When it was conducted in vivo, it was found to be co-recruited on the cellular promoter along with a cellular transcription factor. So the promoter, it got first bound by the transcription factor, and the transcription factor co-recruited this particular protein for binding to the transcription factor. So which one of the following statements seems to be the best interpretation of the above findings? X is a DNA binding protein. It is not. When they conducted in vitro DNA binding activity with protein X, they found out that there was no DNA binding activity identified with protein X. So it is not a DNA binding protein. X physically interacts with the transcription factor. That may be the reason. Why? Because X interacts. When they conducted in vivo experiment, they found out that the cellular transcription factor co-recruited this particular protein X, and it physically interacts with the transcription factor. That is correct. X modifies the chromatin for transcription activation. It doesn't modify the chromatin because it is co-recruited onto the cellular promoter along with the cellular transcription factor. X is a chaperone. That is also not correct. So the answer here is option 2. The next one. Protoplast fusion is used in plant tissue culture for various applications. In protoplast fusion, naked plant cells are used. Yes, they don't have a cell wall. Then transfer of organelles is not possible. That is not correct. Partial genome transfer is involved. Yes, that is also correct. Cells from two different plants can be mixed together and forced to fuse. That is also correct. So which one of the following combination of the above statement is correct? It is A, C and D, option 2. The next one. Following are few statements for regeneration of plants from explants tissues. Explants or tissue. So in tissue culture experiment, they have given some hormones here, and the mechanism of action of the hormone they have asked for regeneration of plants from explants and tissues. Which of the following combination of above statement is true? Okay. Cytokinin is required for shoot development. Of course. So and A is correct. Auxin is required for shoot development, not shoot development, it is for root development. So B is not correct. Auxin to cytokinin ratio is very important. That is also correct. High auxin ratio will induce root development, and high cytokinin ratio will induce shoot development. So that is important. Then jasmonic acid is required for both shoot and root development. Not jasmonic acid, it is auxin and cytokinin that are required for both shoot and root development. So the answer is option 1, A and C. Then next one. Choose the correct sequence of events in a next generation sequencing technology based whole genome sequencing project. I am directly going to the answer. It is option 3 because it is only a sequence of steps required. So it is DNA extraction, then you shear the DNA, then adapter ligation, library amplification, sequencing, assembly, finishing, annotation, and then submission to GenBank. So these are the sequence of events in a next generation sequencing technology based whole genome sequencing project. Then next one. An investigator discovers a new receptor for a ligand and wanted to identify the binding partner of the receptor, that is its co-receptor. The anti-receptor antibody is not available, but anti-GFB antibody is available. Which of the following strategies is most likely to identify the co-receptor? Now, the investigator, he discovers a new receptor for a ligand and he wants to identify the co-receptor. The anti-receptor antibody is not available. So the antibody against this particular receptor is not available, but you have anti-GFP antibody is available. So which of the following strategies is most likely to identify the co-receptor? The GFP receptor fusion protein is expressed in a cell line and analyzed by LC-MS. So here the GFP receptor fusion is expressed in the cell line, but you are not using anti-GFP antibody here, but you have to use that. Only then you will get specificity for your reaction. So one is not correct. The GFP receptor fusion protein is expressed in a cell line and the cells positive for GFP were sorted out, lysed and run on a polyacrylamide gel. That is also not correct. Cells positive for GFP will not always identify your co-receptor. The GFP receptor protein is coated on ELISA plate followed by ELISA with anti-GFP antibody. That is also not correct. The next one. The receptor is cloned as a fusion protein of GFP and expressed in stimulated cells. The immunoprecipitated complex obtained by anti-GFP antibody was analyzed by LC-MS. That is correct. That is more correct. So first what you do, the receptor is cloned as a fusion protein of GFP. The receptor GFP complex you will get as the protein expressed and expressed in stimulated cells. The immunoprecipitated complex obtained by, then you add your anti-GFP antibody, then immunoprecipitated complex obtained was analyzed by LC-MS. So that is more correct. So answer is option 4. Coming to December 2013 question paper. First question. Which of the following microbial fermentation are anaerobic? Ethanol is anaerobic. Acetone butanol also is anaerobic. So answer is option 1. Next one. Encasing of which of the following plant cells in a gelatinous matrix is referred to as artificial seed? So it is somatic embryos that are encased in the gelatinous matrix and they are referred to as artificial seed. Next one. Which of the following transgenic crops have been approved for commercial cultivation in India? So it is cotton, which has been approved for commercial cultivation in India. Next one. Tryptic digest of a heptapeptide built from 3 lysine, 2 alanine, 1 tyrosine and 1 phenylalanine yielded tri and tetrapeptide. Which of the following is the correct sequence of the heptapeptide? So here they have digested a heptapeptide with trypsin and it yielded tri and tetrapeptide. Which of the following is the correct sequence of the heptapeptide? Now trypsin, it digests the carboxyl side of lysine and arginine. K is lysine, R is arginine. R is not there anywhere in the sequence, only K is present. But trypsin is an endopeptidase. It doesn't digest at the ends. So this lysine will not get this lysine and this lysine will not get digested. But here it will get digested because it is inside of the peptide. So the first option you will get 5 and 2 pentapeptide and two peptide, 5 and 2 peptides. Then the next option here also you can see the digestion I have marked over here. Here you will get 2 and here you will get 5 sequences. Then over here the third option, if you see, you will get a tripeptide and a tetrapeptide. So option 3 is the answer. Then next one. While designing an experiment for agrobacterium mediated plant transformation, a student noted down the following points. TI and RI plasmid induce crown gall and hairy root disease respectively. Now what they have asked is which of the above statement is correct. So first one is correct. Enzymes octopine synthase and nopalin synthase involved in the synthesis of octopine and nopalin respectively are encoded by tDNA. That is also correct. All of the 6 Vir genes, Vir A, B, C, D, E, G are absolutely required for virulence. They are not absolutely required. So option C is not correct. Almost perfect 25 base pair direct repeat sequences flanking all TI and RI plasmid in the tDNA region is essential for tDNA transfer. That is also correct. So the options A, B, D are correct. Answer is option 4. The next one. An unknown peptide was isolated from the leaf of a medicinal plant and purified to homogeneity. The peptide did not yield any sequence when subjected to Edman degradation. Now what for is Edman degradation used? It is used for purifying protein by sequentially removing one residue at a time from the amino end of the peptide. For that only you use Edman degradation. However, tryptic digest of the peptide produced a unique sequence. The mass of the intact peptide was found to be 18 Dalton less than that obtained from the trypsin treated sample. The possible interpretation of the above experimental results could be that which of the above statement is true? Now, it didn't yield any sequence on Edman degradation. So this may be due to the circular or cyclic nature of the peptide. So we'll see the first one, that is the N-terminus of the peptide was blocked by acetylation or methylation. They have not told anything like that in the question, whether it is acetylated or methylated at the end. The peptide was cyclic and contained at least one internal arginine or lysine residue. That may be correct. It is cyclic in nature, that is why it didn't get degraded, it didn't get digested on Edman degradation. And it may have one internal arginine or lysine residue. That is also correct. That is why it underwent tryptic digest. Because trypsin cleaves at the C-terminal end of arginine and lysine. The peptide was cyclic and contained a lysine and arginine residue at the C-terminus. That is wrong. If it is at the C-terminus, then tryptic digest would not have happened. The peptide was cyclized by peptide bond formation between alpha amino group and alpha carboxyl group. That is also correct. So answer is B and D. Option 4 is correct. Then next one. A gene is regulated by a novel transcription factor. The following techniques may be used to identify the cis regulatory element in the 1 kb promoter sequence of the gene where the novel transcription factor binds. So, you have got a gene which has got a promoter, and onto this promoter a new transcription factor is binding. Now you have to find out the technique which is used to identify the cis regulatory element. Cis regulatory element is in your DNA sequence. Trans is in your proteins. So cis regulatory element in the 1 kb promoter sequence of the gene. So what are the ways you can analyze and find out? So it is bioinformatic analysis, no. Cell-based reporter assay, no. S1 nuclease assay, no. Electrophoretic mobility shift assay, it can be used, but since it is a 1 kb promoter sequence, you cannot use EMSA because EMSA is used for analyzing protein DNA interaction on a small fragment, that is around 20 to 30 base pairs. But here it is nearly 1 kb, so it cannot be used. DNA is one footprinting analysis. Yes, it can be used because it is used for scanning a large DNA fragment for protein DNA interaction. So answer is option E. E only. Answer is option 4. Coming to the next question. A protein is encoded by a gene which is 5 kb long and has 3 Hin3 restriction enzyme sites. The first one is 0.5 kb from the transcription start site. Second one is 2.5 kb from the first site, and the third one is 0.5 kb internal to the stop codon. So a protein D is encoded by the gene which is 5 kb long. So let this be the tentative map of the gene. So this is 5 kb long and it has got 3 Hin3 restriction enzyme sites. The first one is 0.5 kb from the transcription start site. So this is the transcription start site and it is 0.5 kb from the transcription start site. The second one is 2.5 kb from the first site. So from here it is 2.5 kb. You have got another Hin3 site. And the third one is 0.5 kb internal to the stop codon. So here you have the stop codon and it is a 0.5 kb internal to the stop codon, that is over here. From here to here it is 0.5 kb. The second site is polymorphic, this one. This site is polymorphic. In order to find out whether fetal cells contain the normal or the mutated gene, total genomic DNA from the fetal cell was isolated, completely digested with Hin3, separated in an agarose gel, transferred to membrane and detected by a probe against the region between the second and third restriction site. So you want to know whether the fetal cells contain the normal or the mutated gene. Mutated means here what they mean is whether it is, because they have already told the second site is polymorphic, so they want to know whether the second site is mutated. Okay. And so what they did was they isolated the total genomic DNA from fetal cells, digested it with Hin3, separated them on an agarose gel, that is they did southern blotting, transferred to membrane and detected by a probe against the region between second and third restriction site. So this is the probe region over here, 1.5 kb. Okay, this is the probe region between second and third sites. This region is 1.5 kb. Then which of the following band patterns will be obtained if the fetal cell is heterozygous? Now consider the condition of a heterozygote. So heterozygote, it has one allele which has all the three sites intact and functional. So this is the heterozygote. One allele, it is all the three sites, 1, 2 and 3 Hin3 site, they are all intact and functional, while the another allele contains a site which is defected or polymorphic. So this allele, it will contain the site which is defective or polymorphic, this site. This is the polymorphic site, it will be defective because in that site the Hin3 restriction site is mutated. So it will be polymorphic and it will not get cleaved upon treatment with Hin3 enzyme. Now the probe is 1.5 kb, it's already been told in the question. Detected by a probe against the region between second and third restriction site. So it is 1.5 kb and that is the probe region. Hence only so wherever this probe region is complementary, only those region will be visible on the gel. The whole DNA band 5 kb region will not be visible on the gel. So here, if it is a heterozygous, you will get two bands. One will be 1.5 kb from here to here. This region doesn't have the probe recognition site, so it will not be detected. This 0.5, 2.5, 0.5 kb will not be detected because it is the region which is not complementary to the probe. Only this region is complementary to the probe. So a 1.5 kb band will be detected. Here it will not get digested with HIN3 because it is a polymorphic site and it got mutated. It is a total, it is 4 kb long. So here it will, in this region the probe will bind and you will get a 4 kb band also. So the answer here is option 2, that is 4 kb and 1.5 kb. That band will be detected. This one. Then next one. Given below is the experimental protocols to find out the exact location of a repetitive DNA sequence in mitotic chromosome by fish fluorescence in situ hybridization. Which one of the protocols will give the correct result? So this is just a sequence. They have asked for the protocol and the answer is option 3, that is mitotic chromosome were fixed on the glass slide, then they were denatured, incubated with biotinylated satellite DNA, incubated with fluorescently labeled avidin, localization observed under fluorescent microscope. So answer is option 3.